So the next test that we're going to do is a rapid susceptibility test called the cephinase test or the beta-lactamase test. So we're going to use a disc that is impregnated with nitrocephin, and this will allow us to test for the enzyme beta-lactamase. This beta-lactamase enzyme, it makes, when it is present in a microorganism, it shows resistance. The organism shows resistance to penicillin and some of the cephalosporins. Okay, so we're looking for a positive test will indicate that a bacteria might be resistant to certain beta-lactam drugs that are used to treat infections. Okay, and once again, this disc looks very much like the Kirby-Bauer discs. Okay, so it has a little beta on it. I'm going to, with the sterile forceps, I am going to place 2 discs on a clean slide. And then I'm going to moisten. I'm going to moisten with sterile. I'm going to moisten with sterile water. And I want to be careful not to overly moisten. So I'm going to put a drop. Okay. Just notice that I accidentally picked up 2 discs here. There we go. So I want to moisten the disc. Okay, you can see the discs are carefully moistened, but not overly so. Okay. What we're looking for is a color change. Okay, we want a color change to pink. So I'm going to test, first I'm going to test against my, this is a Staph aureus. And all I'm going to do is I'm going to rub that colony on that disc that has the nitrocephin. Oops. Okay. I'm going to let that sit for a few minutes, okay? Next I'm going to test another organism. And this is a Moraxella catarrhalis, okay? Which you remember, it's similar to the Neisseриas, okay? And same thing, I'm going to, you can take a couple of colonies. I'm going to inoculate that on the top of that moistened nitrocephin disc. And what we're looking for is we're looking for a color change, pink to red, okay? Well, you can see that you should be able to see already that the Moraxella is showing a color change to a dark pink or to red, indicating that Moraxella is beta-lactamase positive. A beta-lactamase positive organism represents a likely resistant to the penicillins, which means that would not be indicated, those antibiotics would not be indicated for this organism to be tested, to be set up to treat that patient. The staph at the moment is still negative. Typically you would hold this 5 to 10 minutes, likely this one's going to stay negative. Most of their staphs are typically negative, but they can be, whoops, that's my duplicate disc, uninoculated. So it's possible we'll hold on to that beta-lactamase. It could turn positive, but at the moment you can see that the one on the left is positive and the one on the right is negative so far. Okay. Okay, the last test that we are going to do on this virtual lab is the penicillin binding protein assay. Okay, so the PBP2A. Okay, this is penicillin binding protein assay looking for methicillin resistance in Staph aureus. Okay, so this means we have to have a known, we start with a known Staph aureus is what we start with to test to test these organisms. Okay, there's no reason to test anything else other than our staph. All right. So once we have a Staph aureus, we really want to know if that organism is going to be positive for PBP2. This allows us to report a rapid methicillin resistance, and that's very helpful because that means as soon as we can give this rapid result, that allows the physicians to know whether or not they can continue or they need to adjust the treatment that they've started on any given patient. Okay, typically, particularly if this is in like a sterile site, maybe a blood culture or sterile body fluid, they might start with vancomycin, which is fairly toxic to the system. But if it's a methicillin resistant staph, they don't have a lot of other options. So they may go ahead and start IV therapy with vancomycin to make sure they can hit that organism and treat it, get it under control. However, if the methicillin, if it's methicillin susceptible, that means that they have many more options for treatment. So if we can allow them to know pretty quickly, within about 5 minutes, whether or not they can treat with other antibiotics, or they have to use some of the more the harder, the hardier, that basically they can kind of hit hard with the certain antibiotic, but it might be harder on the system. Well, for the most part, they want to make sure that they are treating this staph correctly. Okay, all right. So this is a really, along with a lot of these other kit tests, that's what this is. This is a kit test. We have 2 reagents, and they allows us to, once again, we're going to add reagent 1. We're going to inoculate our test organism, and then we're going to add reagent 2, mix it a little bit, and then, like I said, this is a immunochromatic membrane assay, which we use quite a bit in the lab. We use these different varieties of these immunochromatic assays. This particular one uses monoclonal antibody fragments, and these fragments will detect the PBP2 protein directly from bacterial isolates. So the presence of the PBP2 protein indicates the organism is methicillin resistant. Okay, so this test allows us, we don't have to do a full susceptibility test. It allows us to test for these antibody fragments that are present in methicillin resistant organisms. Okay, we're going to start with the staph that we actually tested for our cephanase, this ATCC known susceptibility, known strain. We're going to drop 2 drops of reagent 1. We're going to pick up 2-3 colonies. We're just going to make sure that is released in that tube.
Oh, sorry.
Then we're going to go ahead and we're going to drop 2 drops of reagent 2. You want to make sure that the color changes from that blue to now it should be clear again. And now we're going to drop that immunochromatic strip directly into that tube. And this takes only about 5 minutes. So you are going to see if you can see this. Okay, the liquid is slowly going up the strip. As with all of these kind of immunochromatic tests, it's going to have a control line and a test line. The control line should always be positive. If it's not positive, the test is invalid. So while that is incubating, we're going to test another strain of Staph aureus. I'm going to do this the same way. We're going to drop 2 drops of our reagent 1. This time we're going to use this ATCC 4330. Once again, these are known Staph aureus strains. And we want to make sure we get all of that colony off into that reagent. Two drops of reagent 2. Remember, we want to make sure that changes from the blue to the clear. Okay, and now we're going to drop that strip as well. I always like to make sure while it's incubating that right away I want to see the movement of the liquid going up the strip. You can just see it moving, so that's a good thing. Now already in my first staph that I tested, I can see the control line is positive. That's a good sign. So far, I only see the control line positive. I do not see a second line test line, okay? But it's good to know that the control line has worked, okay? Kind of taking a look at this. Kind of waiting for this. Still moving up the strip, which is good. Okay, going back to my staph, my beta-lactamase, do you see that that staph that we tested is now also positive? So it really does, that beta-lactamase test, you really do need to hold these, sorry, let me get that so you can see it. You really do want to hold these for that entire incubation time, because organisms like the Moraxella, sometimes they can go rapidly positive. Sometimes the organisms such as Staph aureus, takes a little bit longer to go, but it indeed also was positive. So both of these organisms were beta-lactamase positive. Okay. That is not unusual at all, because a lot of the staphylococci have become resistant to penicillin a while ago, so that the straight penicillin is not often is not used to treat staph infections at all. All right. Okay, looking at my second staph. Okay, I think it's kind of hard. Let's see. I think, can you see that there's a strong control line, but there is also now a second line that is coming up. It's a little faint right now, but I think that'll change. I think that'll get, oh, whoops. That happens sometimes too. Playing around with my test system. Okay, you can see there that the test line is on the bottom. So my control line's on the top, my test line is on the bottom, and that is positive. So this is positive for PBP2, meaning this organism is likely to be methicillin resistant, okay? So that's a rapid test that we can do some susceptibility testing for, and then following that up with our commercial system that allows us to do a full panel of antibiotics and determine what the actual MICs would be for those. So all of these tests we did are ones that we can do mostly on the bench. Some require incubation overnight before we interpret them, but some of them are done within just a few minutes and can give us rapid information that can be helpful to the physicians for treatment. And then, of course, typically these would be followed up with a more robust testing of multiple antibiotics as well. Right? Okay, well, that is the end of our session on susceptibility testing, and at least for the rapid testing that could be done in most clinical labs. And if you have more questions, I hope you have them ready to discuss.